Hello, everyone, and welcome to today's webinar, High-plex Spatial Proteomics Workflow for Enhanced Breast Cancer Research. I'm Christina Jewell of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Thermo Fisher Scientific. To learn more about our sponsor, please visit thermofisher.com. Now, today's event is interactive, and we encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We will answer as many of your questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. Now, without further delay, I would like to now introduce today's speaker, Nancie Mooney, Scientist, R&D, Protein and Cell Analysis at Thermo Fisher Scientific. For a complete biography on Nancie, please click on the Presenter tab at the left of your screen. Nancie, welcome. It's so wonderful to have you here today. You may now begin your presentation.
Hello, I'm Nancie Mooney. I'm a scientist in research development and protein and cell analysis for Thermo Fisher Scientific. And today I'm going to be presenting on high-plex spatial proteomics workflow for enhanced breast cancer research. Essentially, how to put together the experiment so that you can produce images that look like this. Spatial biology is a rapidly growing field of study that explores how cells and molecules, like biomarkers, are distributed within and interact within tissues. Spatial biology, as it is evolving, is starting to require higher plex to enable whole tissue imaging and complex phenotyping of cells. And so really this requires biological samples that you can sample process appropriately while using the appropriate kinds of detection reagents, fluorescent dyes if you're using fluorescent imaging platforms, data analysis, and of course the expertise and knowledge it takes to put all of those together. So today I'm going to be talking about what it takes to put together a workflow to create images like this one here in the center, using the Invitrogen Alora spatial amplification reagents and the new Invitrogen EVOS S1000 spatial imaging system. So prior spatial biology techniques were lower plex. So for looking into, say, invasive ductal carcinoma of the breast, and these 8 markers like vimentin and various CD markers to look at the immune cells within this tissue, if you were using kind of the original in the space, you'd be looking at DAB, which is an HRP colorimetric stain. So by using DAB, you could look at the localization patterns and the overall signal intensity for the most part of these various markers. But it would be very hard to make any kind of statements about co-localization based on this kind of staining. You might be able to make some assumptions based on sequential tissue slices and such, but you can't use multiple DAB stains on a single slice of tissue. As the technology progressed, we started getting into fluorescent immunohistochemistry, where you could use multiple fluors on a tissue, but without some kind of spectral and mixing to account for spectral bleed through between fluorescent molecules. You had to be very mindful of the fluorescent spectra of the fluorescent dyes that you were using to ensure that the targets and the staining were unique and had no bleed through that could confound the staining. So you could do something like a 3 or 4-plex, maybe up to a 6-plex if you had a very complex microscope, which allowed for more plex and therefore more complex multiplexing. So you could say stain for 3 different CD markers against markers that are expressed for T cells, for example, CD3, CD4, and CD8, to start looking at cytotoxic and helper T cells within this tissue. And by looking at the co-localization, you could start establishing the different kinds of populations. Or looking at things like proliferating cell markers like PCNA and vimentinoprohibidin, which are epithelial matrix markers within this tissue to tell a different part of the story. But again, you have to do careful planning and monitor your fluorescent spectra and your targets to be able to tell these stories. With these new technologies, however, like the Alora spatial reagents and the S1000, we can multiplex up to 8 biomarkers into a single tissue and then include DAPI, allowing us to build up a 9-plex. So in this invasive ductal carcinoma, I was able to take those 8 prior biomarkers and put them into one sample. Here I have a video showing the various markers being turned on and off to highlight the multiplex we were able to enable. So this is that same image, but in stasis. So here we have our unmixed merged composite where we are able to take our 8 different biomarkers and DAPI and multiplex them into a single image. Over here, we have the individual channels in the various colors as single targets. So you can see here we've got vimentin, various CD markers, and so on. And here you can see that the individual channels. So the EVOS S1000 also enables whole slice imaging. So here we've got the invasive ductal carcinoma in a tiled image. And again, having the targets turn on and off to kind of highlight the multiplex that we were able to enable. You can see here that spatial biology really enables us to highlight the heterogeneity of this tissue. So if you were going to do some kind of science that didn't involve some kind of spatial component, you would lose a lot of this information that could be critical to understanding the biology of this invasive ductal carcinoma. So this is that same multiplex, but again, in static. Again, you can see the heterogeneous nature of this tissue and how when you've got all of these targets, you can really start calling out these interesting patterns. So when it comes to multiplex, it's complex. There's multiple aspects to consider. For example, as I called out before, all of this. Nevertheless, how the various detection modalities might interact with each other or how to choose appropriate treatments in order to enable these various modalities. So how you prepare your sample, the antigen retrieval, how you might block various backgrounds, both autofluorescence and just simple protein backgrounds, as well as how you're going to detect the target and how you might detect multiple targets. In many cases, the order matters. So how you might block or antigen retrieve for one modality might interfere with the next. And so really this complexity is less linear and more exponential in a lot of ways, kind of a checkers to chess to 40 chess. So in this webinar, I'm going to talk to some of these considerations that you need to make these big multiplex samples. So first off, biological samples. So you want to consider your tissue type and how it's prepared. So tissue thickness is something that one needs to take into account. For the most part, we've been using standard 5 to 8 micron slices. However, there can be slices that are thicker depending on the tissue type, especially. If you have thicker tissue, you may need to incubate some of your reagents for longer. It's also important to consider tissue makeup. If your tissue is, say, normal lung tissue, which could be relatively diaphanous and therefore fragile, you might want to consider your detection modality and whether or not the tissue can hold up to what you're doing to it. In other situations, you need to consider the autofluorescent characteristics. Is it blood rich or really strong in collagen? If there's going to be a lot of autofluorescence, you might want to consider some kind of signal amplification so that you can bring the signal of your biomarker up above this autofluorescent noise. Fixation type, of course, can change the amount of autofluorescence and how your tissue can be handled as well. In this situation, I use formalin-fixed paraffin-embedded tissues, though cryopreserved tissue could also probably be used. It can be more difficult to multiplex with cryo tissue due to the fragility compared to FFPE tissue, but everything needs to be optimized in the scientist's hands. As I said before, I use formalin-fixed paraffin-embedded tissue in the workflow that I'm going to be talking to today. When it comes to sample processing, are you going to be using an automated or a manual system? In this case, I used a Leica Bond automated slide stainer, an RXM system actually. So the nice part about the automated slide stainers is that they can be consistent and efficient, and they have standardization and help reduce human error. However, they can be pretty expensive, and as a complex system, sometimes there's some complications there. Manual processing, of course, can lower costs, especially in terms of initial costs, and that it's way less expensive. You also have more flexibility and control over your experimental setups as you kind of figure out what works best in your hands. You also have less dependent on complex equipment. So whichever system you use can be enabled for the Allura and the S1000, but often automated staining is better for high multiplex experiments just because of the complexity and the time that it might take. So when it comes to your detection reagents, the workflows that I'm going to be talking to today are based on primary antibodies. So if you don't have good primary antibodies, it doesn't matter how good the detection system is in terms of laying down your floors or how wonderful the microscope is. If your primary antibody isn't specific or clean, there's nothing in the world that can save your science at that point. Garbage in, garbage out in many ways. So it's important to use validated primary antibodies that you can rely upon. Thermo Fisher, of course, has our advanced verification antibodies, but really just make sure that it's an antibody that you're comfortable with and you do the appropriate controls and optimization. If you don't optimize your science ahead of time, you're in for a lot harder work when it comes to data analysis and imaging. So when it comes to detection reagents that are based in primary antibodies, there's a couple different options. For the workflow that I am talking to today, I use the Alora spatial amplification, but that's not the only system that you can use. There are primary antibody conjugates, which are primary antibodies that have the fluors directly conjugated to the primary antibody. And those are wonderful for a lot of reasons. It can mean that you can multiplex 8 biomarkers in a single tube of antibody and apply it to a tissue in one step, giving you a lot of speed in your experimental setup. However, it can limit you a little bit in terms of flexibility. You either have to conjugate the antibodies yourself or buy antibody conjugates. And sometimes it can be difficult to find one in all the colors that you want. If you've already got a fridge full of antibodies that you know work, sometimes doing the antibody conjugations are a little complicated. You can also use primary secondary antibody detection, but then you have to be very mindful about species. If you have multiple mouse antibodies that you want to use, that might be difficult because the secondary fluor that you use to detect your primary antibody will detect both mouse antibodies, causing cross-contamination and problems with your signal. The Alora spatial amplification can use any primary antibody that is mouse, rabbit, or biotinylated, giving you high flexibility. But because it's an iterative labeling strategy, it takes a little bit much longer than the primary antibody conjugates. So these are things to consider when you're deciding on your detection system. The other thing to consider is the detection threshold and your target abundance. So if your target isn't very abundant or has very diffuse staining, primary antibody conjugates might not be able to deliver the signal to background that you need to bring that target above your detection threshold. In that case, Alora spatial amplification reagents might be exactly what you need, because the signal amplification that it takes when we lay down these additional fluors bring the overall signal above that detection threshold. So Alora spatial amplification is a tyramide-like chemistry. So it relies upon primary antibodies and HRP enzyme. What you end up doing is you lay down your primary antibody on your tissue against, again, your antigen of interest. You then go in and detect it using a secondary HRP antibody or a streptavidin HRP if you're using a biotinylated primary. You then use the HRP that's associated with that secondary to activate the Alora reagent, causing it to covalently bind to the tyrosines near your antibody detection structure. Again, since this is covalently bound to your tissue, at which point you can strip away the primary and secondary antibodies. In this case, I used heat and citrate buffer. Once you remove the primary antibodies, because the Aloras are covalently bound, they remain. Then you can iterate and go back to stage 1 with a different Alora reagent, enabling multiplexing. So like I stated before, there are 8 different Alora reagents in 8 spectrally unmixable colors. These have been optimized to multiplex well together. As long as your antibody is rabbit, mouse, or biotinylated, you can use this Alora kit to lay down these Alora dyes covalently to your tissue. And as I stated, these Alora reagents have been optimized to work together so that you can multiplex with 8 different biomarkers as well as DAPI. So to create the images that I've been showing in this webinar, I use the EVOS S1000 spatial imaging system, where we're really looking to simplify spatial biology and the imaging that it requires. The EVOS S1000 allows for spectral and multiplexing up to 8 protein targets and DAPI. It creates high-resolution images using a Zeiss APO objective and a CMOS camera. The microscope has a 4-slide capacity and has custom IR laser autofocus that enables whole slice scanning. So that image where I showed the entire slice of the invasive ductal carcinoma all imaged together, this autofocus allows that to take place because it can keep all the tissue in focus for all the images that takes. These images are in OMI-TIFF file format so that it's easy to export those images into data analysis software like QuPath or Indica Lab Halo. And the instrument has intuitive software controls that make it really straightforward to create these images. All in all, the instrument is designed to make spatial biology easy and streamlined. And I'll talk a little bit more about the workflow later in this webinar. So let's talk about how I produced this image in specific. So this unmerged composite with these 8 biomarkers plus DAPI. So first thing we need to think about is what targets we want to do. So do your research, consider what you're looking into. In this case, I was looking at breast cancer markers. You want to consider your antibodies and samples, like I spoke to earlier. Once you've got your antibodies in your samples, the next thing you want to do is create single target controls of each of your antibodies. This is to optimize your primary antibody staining. This is really important when it comes to unmixing, because if you have clean antibodies, primary antibody staining for each of your targets, it makes the data analysis and unmixing of your multiplex much, much easier. Optimizing in this step is the best way to end up with good data at the end of the day. So for the singles target controls, you're looking for bright, clean staining with no nonspecific staining, and of course, appropriate localization. Once you've optimized in this step, you can then create your multiplex. Some things you want to keep in mind for your multiplex setup is target localization versus spectral overlap. So if you are expecting your targets to co-localize, for example, if you have multiple targets you expect to be in the nuclei, you want to make sure that those targets are not in fluors that have as much spectral overlap. This makes the unmixing more complicated and can make the data more difficult to rely upon. So if you have multiple targets that are nuclear, I strongly recommend putting them in spectrally dissimilar fluors. So don't put all your nuclear targets in 488, 514, and 555, which, as you can see here in the spectrum, have a lot of overlap. Same with 647 and 700 here. The other thing to account for is target staining and target abundance versus the brightness of the dye. Though the Allura reagents have been optimized to multiplex well together, we're still dealing with different fluors and different channels. So we have some that are brighter and some that are slightly dimmer. If you have a target that's less abundant or more diffuse, you might want to put it in a brighter channel, like 488, 647, or 594. If you have one that's much brighter or more abundant, that's a good target for 700, 750, or 430. The other thing to account for is that the abundance of the target and the Allura channel that you place it in can change the amount of primary antibody that you actually need. So if you optimize using your single target controls in one color, you might need to add more primary antibody when you switch it to one of the dimmer channels. For the most part, this follows this trend. So if you use 1 to 10,000 of an antibody in 48, 594, you'll probably need something more like 1 to 1,000 to get the same kind of staining in Allura 430. So for the experiment that I showed earlier with the invasive ductal carcinoma with kind of an immune oncology panel, I did some research, read some papers, and then looked in the fridge to see like what antibodies I already had. I ended up purchasing a few more antibodies and then made sure that my samples were in good stead. Then I ran my single target controls, looking for bright, clean, and appropriately localized staining for each of these antibodies. I ended up starting at 1 to 1,000 for all of these antibodies and then titrating down over time, which I'll show in the next slide. I then looked at the localization and what I expected to co-localize and assigned them to the various Allura channels. Again, accounting for localization and the brightness versus which Allura channel I wanted to put each of my targets in. So in terms of my single target controls, of course, I optimized my primary antibody because this is a key step. So in this case, I'm showing CD4 and CD68 at my initial concentration, 1 to 1,000. And you can see that I'm seeing some off-target kind of blurry effects. This is a strong indication that I had too much primary antibody and that the signal amplification was bringing up non-specific staining. So in both cases, I brought my antibody concentration down to 1 to 5,000, which got rid of this non-specific target or this non-specific staining and really sharpened up my staining significantly, as you can see here. Once I was happy with the antibody concentration and the kind of staining I was seeing in my single target controls, I then laid them out and started looking at the localization and kind of organizing them overall. So based on the phenotypes that I was expecting based on these targets, I knew I should see co-localization from CD3 with CD8 and CD3 with CD4, and not so much in the other channels. So when I was putting together my multiplex, I took that into account. I knew that I would want to put these in spectrally distinct channels with very little overlap. In my other channels, I wasn't expecting to see as much co-localization, so I did not need to be as cautious with these channels. In putting together my panel, as you can see, I've got my CD markers that I expected co-localization in different parts of the spectrum. So I've got one in 488, one in 594, and one in 700. So none of them are spectral neighbors to one another. Another thing that I needed to account for was the fact that when I was doing my single target controls, I used Alora 594. And so the optimal concentration for 594 was 1 to 10,000. However, when I decided to put Vimentin as a strong stain with an abundant protein in Alora 430, I increased the amount of primary antibody concentration from 1 to 10,000 to 1 to 1,000 to account for the slightly dimmer signal that I see in 430. This became my multiplex panel. And that is how I got this image. Again, with the 8 targets that I've been looking at in their various Alora's channels, and then again, showing you as the targets toggle on and off. Okay, so how did I do this imaging and then the unmixing? So for the EVOS S1000 unmixing workflow, you need not only your multiplex with all the fluors that you wanted, you need single color controls. In this case, that means you need an unstained control slide that has no fluorescent staining on it whatsoever to account for any autofluorescence from the tissue. And then for every fluor that you have on your multiplex, you need a single color control for that. So for example, in this situation where I had DAPI and all 8 Aloras, I have a DAPI alone slide. Then I have a slide that was only stained with Alora 430, 488, 514, etc. Once I had all of those slides, I went to the S1000 system. First, I put in my multiplex to set my exposures to give me kind of a ground truth of what kind of fluorescent intensity I was expecting. I then went through the unmixing matrix setup workflow. What happens is you put in each set of the single color controls and take a set of images. These images are then built into the unmixing matrix that involves some math about which fluor contributes the most in each of these channels, which are kind of a mixture of the channel and the filter set and the LED that's used to create it. By using this unmixing matrix, what one can do is you basically have pixel by pixel, the machine is able to look at the spectral signature in each pixel and then know how much of that fluorescent or how much of that signal came from each of the fluorescent profiles of the fluors that you've fed into the system. For more information on this, I strongly recommend going to the EVOS S1000 working guide or on thermo.com spatial biology. They have more resources there to better understand this part of the science. In any case, once you've got your unmixing matrix, what this allows is that the S1000 can now unmix each of these images and remove spectral bleed through from each of the fluorescent channels from one another. So with the raw calibration images, you can see that there was a lot of bleed through from 514 into other channels. However, post unmixing, that bleed through is eliminated. You can also see that over here in 647, 700, 750. This is also cleaned up in the post unmixing calibration. So this unmixing allows us to take fluors that have fluorescent spectral overlap and clean them up enough to use them as individual targets in our experiments, thus allowing us to create images that look like this. So once you have data like this, you can then do data analysis for cellular phenotyping and other kinds of spatial biology. In this case, I used Indica Labs' HALO program. And so I took a selection of my invasive ductal carcinoma stained with the target that we were speaking to earlier. I ran a cell segmentation analysis with the phenotypes that I called out based on the targets that I knew were in the sample. With this segmentation, I was able to get percentages based on which types of immune cells and what proliferating cells were in the sample. So here we are able to see that there were 23% proliferating cells and various percentages of immune cells, including B cells, macrophages, cytotoxic T cells, and helper T cells. Furthermore, I was able to use the HALO program to distinguish between the Vimentin positive and lightly overexpressing regions versus Vimentin negatives, kind of low Vimentin regions. Once I had that segmentation, I was able to put together this graph showing that where the localization of the immune cells were in this invasive ductal carcinoma tissue. As you can see, we were able to show that these immune cells strongly co-localize to the Vimentin positive regions of this tissue and do not appear as much in the Vimentin negative regions. So of course, I wanted to show that it's not just the targets that I showed that work in this particular workflow. So this is that same invasive ductal carcinoma, slightly different region, or on a different slide, where I was able to run a completely different multiplex using the same workflow. So in this case, this is more of a breast oncology panel where I was looking at E-cadherin, estrogen receptor, mucin, progesterone receptor, HER2, Ki67, smooth muscle actin, and P53. As you can see, again, I am able to have 8 distinct biomarkers in all of these channels and really call out the heterogeneous nature of this invasive ductal carcinoma and show localization of these targets as they compare to one another. I also wanted to show that this workflow doesn't just work in cancer tissue. So here's an FFPE mouse brain slice that I stained with various neuronal markers. Again, highlighting that this workflow is very flexible and can be used for all manner of antibodies and tissue. So hopefully during this webinar, I was able to give you some good insight about how to put together these kinds of samples and run this kind of imaging. However, I know that it's always nice to have more resources. So of course, as I mentioned before, thermofisher.com/spatial biology has a lot of different resources. We have webinars, protocols, technical guidance, and more. You might have actually found this webinar on this webpage. We'll be putting out more app notes as time goes on. And we really hope that this resource is helpful in your spatial biology experimentation. So in conclusion, I was able to use the Alora spatial reagents to create this, to stain this invasive ductal carcinoma with 8 different biomarkers and then run an analysis. So the Alora spatial reagents are 8 spectrally unmixable colors that can iteratively and sequentially covalently label target proteins on your tissue. This produces robust and stable staining on your tissue. Using your own primary antibodies as long as they're mouse, rabbit, or biotinylated. You can use low concentrations of primary antibody from 1 to 1,000, even down to 1 to 100,000 to detect nearly any target abundance in your tissue on nearly any tissue type. I then use the EVOS S1000 spatial imaging system to spectrally unmix these 8 protein targets and DAPI using the Zeiss high-resolution objectives in the CMOS camera to get good image quality. This has a 4-slide capacity and custom IR laser focus that allowed me to take whole tissue slice images of my samples. These were then produced as OMI-TIFF that I could feed into a data analysis program and enabling me to do cellular phenotyping and other data analysis, all the while using intuitive instrument controls to make getting these images very easy. So hopefully this webinar was very helpful. I want to thank you for all of your time, and I hope this was helpful. Thank you so much.
Nancie, thank you so much for that excellent presentation, and welcome back on camera for our live Q&A. Now, we do have some interesting questions that have already come in, but to our audience, if you haven't already done so, please do so now. Submit those questions into the Ask a Question box. Click Submit, and we will answer as many of your questions as we have time for at the end of the presentation, which is right now. So, Nancie, let's dive in here. I'm going to go right with this question first. How can high-plex spatial proteomics be integrated with other omics approaches, techniques to provide a more comprehensive understanding of breast cancer?
Sure, absolutely. Great question. So as we build out this spatial biology, especially these big omic technologies, we're really coming to understand more that you really get, it's wonderful to be able to start from the very beginning and walk all the way through the how these cancers kind of put themselves together. So being able to match things like transcriptomics, like metabolomics, as well as proteomics, you start being able to really build out a larger picture and better understand like the underlying mechanisms. So I really find that, especially in spatial biology right now, we're getting really fantastic and interesting data out of the kind of interplay between transcriptomics and proteomics. So you can start looking at what the cells are trying to make and how they're kind of ramping up to produce more of various proteins, and then how that actually ends up in the tissue. I'm also finding that having the different kinds of technologies allow you to do something like transcriptomics often allows you a much higher plex, like you can do near full transcript level transcriptomics of a protein, or sorry, not of a protein, of like a cell or a tissue. And then you can use that data to start digging down to find like individual proteins that you're more interested in based on their overexpression like profiles. And so I think that it's a really kind of interesting way to not even just expand the workflow, but like better understand the underlying science and give you a chance to like hone in on a specific target within a specific, more specific question, kind of giving you like the overlaying full environment and then going to the specific questions of your research.
Thank you, Nancie. Now, do you have any resources available online for the Alora spatial amplification protocol?
Yeah, so as I was saying there, we've got this thermo.com spatial biology. A lot of this stuff is linked there. So we have a user guide, so the actual like how to use the Alora specifically, like what reagents to put in what tubes. But we're also putting out like basically more like the website guide version of this webinar about like things to consider in terms of target selection, how you put the experiments together, troubleshooting, that sort of thing. So those can all be found through the spatial biology website. And like I said, we're going to be updating that a lot. We're really excited to start highlighting to people like the science that we're able to enable and how it can help people's research.
Excellent. And I do want to bring our audience attention to the bottom right corner of your screen where you will see that website, thermofisher.com/spatial biology website. So you can copy that, paste that into your URL. Let's go to our next question here. Is there a difference between Alexa and Alora dyes, the TSA versions?
Sure. So SuperBoost is our TSA, the Alexa-based tyramide amplification. Those were really best optimized on cells. The Alora spatial amplification reagents, we were using that to really optimize for tissue and bigger multiplex. So we wanted to make sure that all 8 of the Alora dyes worked really well together in a way that the SuperBoost TSA, we don't have all of the spectral colors right now. And while they work perfectly well in tissue, they were definitely optimized more in cells. So the new Aloras are definitely meant for your tissue work and this new spectral spatial biology.
Excellent. Thank you for that clarification. Can the 8 dyes be removed from the tissue for reuse with another set of 8 distinct antibodies?
Sure. So for the Alora spatial amplification reagents, no, they are covalently bound to your tissue. So once you put down the 8 fluors, they will be there. This has some, so you can't do any kind of cyclical staining, kind of like some of the other platforms. However, it does mean that you can take these slides, put them in the fridge, and be able to come back to that same slide and see the same level of staining over time. So you have that kind of archivability. So you can kind of come back to the same slides. But no, you can't like strip them off and then reprobe. They are covalently bound and they bind strongly. They're not going anywhere. As long as you've got your tissue, your fluors will still be there.
Thank you, Nancie. Now, is there any information on which filters should be used with the Alora dyes, with the Evo system or with any other imaging equipment?
Sure. So for the EVOS S1000, it has a slightly different setup than the traditional filter set. Because it's a spectral imager, it actually takes many more channels than your core channel in order to support that spectral and mixing. So what happens on the EVOS S1000 is when you're feeding in your single color controls, you tell it the names. Basically, you input what fluors are on your sample. So we'll have like all the Aloras or the Alexas or some of the other standard fluors already built in, and then you give it its spectrum with the single color controls. If you're going to be using the Alora dyes on something like an EVOS M7000 that's actually got more of the cube-based filters, all of those match to pretty standard fluors. So like Alora 488, you use the GFP filter, and so forth. And that is all in not only the Alora user guide, but I think we also will have that in Spectral Viewer and some of the other helpful guides in terms of how to match filters to the various fluors.
Thank you. Now, can you elaborate a little bit on the stability of the Alora dyes compared to the Alexa dyes?
Sure, both are really stable, especially in terms of comparing the tyramide and tyramide-like reagents. So in terms of this super boost versus the Alora, both are very stable. Again, we're definitely helped by the fact that it's signal amplification. So we're laying down many fluors for each like antigen of interest. We've been able to store these samples and rescan them months later and still see very good staining levels. So we're very happy with the stability in it. We really think that scientists will be happy as well.
Excellent. Now, we have an attendee asking if there's a specific resource page on the Thermo site for the EVOS S1000. They weren't able to find it just by a quick browsing.
Sure. So because of these are all very new, like launched within the last couple of months, it's absolutely possible that some of the websites aren't all the way up in the full way that they should be yet. I am in R&D and not the website, so I can't promise anything. But going back to the spatial biology site, if you keep that bookmarked, I'm sure updates will be posted there on the regular.
Excellent. Thank you. And we do have some scientists in our studio with us that just let us know that thermofisher.com/EVOS 1000 is where you can go to get that information. So thank you for that. I've also put that in the bottom right corner. Broadcast message will pop up for you right there. All right. So moving on to our next question. Let's see. Is there any particular benefit of using the EVOS S1000 system versus, say, the Zeiss Axio scans? In other words, is it filter set that is pre-configured for these fluors or filters, and are they customizable for the EVOS S1000?
Sure. So I'm not as familiar with the Zeiss Axio scan, so I can't speak to so much about the comparison. But again, the EVOS S1000 system is set up to be a spectral imager. So it doesn't have, so it has a combination of filter sets and LEDs that it uses in different combinations to basically take, it's not, in, this is the R&D scientist, not the spectral unmixing mathematician. So again, please find the other resources as well, but let me take a stab at this. So the EVOS S1000 is a spectral imager, which means that it's not just taking the single picture of the fluor of interest, it's also taking images in different parts of the spectrum to enable the unmixing of the spectral fluors. So it's taking like a full fingerprint, not just one image in each. It often takes 3 or 4 images per each fluorescent channel to enable this unmixing. And so the filter sets it's not the filter sets that are pre-configured for each of the fluors, it's the combination of the channels and those filter sets to better, again, take that appropriate fingerprint to enable the unmixing. Right now, the system is preset for a certain number of fluors based on kind of the common fluors in the market. And then I can't speak more to how one would use it for different fluors than, say, the standard Alora and Alexa, but they have more resources at the S1000 site.
Excellent. And Nancie, can one reuse the Alora stained tissue with other technologies like IMC?
So I haven't had a chance to prove that out as much, so I'm a little hesitant to speak to it. The Aloras do take multiple round of basically antigen retrieval to strip the fluors off. So depending on the sensitivity of your follow-up detection strategy, you may need to further optimize. But again, the Alluras are covalently bound to the tissue, so they'll survive many things that you would do to them in order to enable other detection modalities. But I just haven't had the opportunity to try different kind of even bigger, more diverse kinds of multiplex at this stage. If you get it to work, let us know.
Yes, please. So another question that came in, it says, is there a dedicated page on Allura dyes specifications and spectrums? And I'd like to let them know that they can go to thermofisher.com/fluorophores to see all of the dyes that Thermo Fisher has, including the Allura dyes. And there they will find links to the Allura dye pages. So I'm going to go ahead and put this link also for you in the bottom right corner. And there we go. Nancie, did you want to comment on that by any chance?
No, that's perfect. I know that we have all of the spectrum in specifications, and then as to which specific website, good news, we've got that for you as well.
Excellent. Thank you, Nancie. Now, we have a few questions in the Q&A box that I'd just like to have you peek at. They may require some additional information that we can reach out to our audience members directly.
Yeah, so I would say that let's reach out to this person offline to get more information. I haven't had an opportunity to use the Alluras on these particular tissue types, but I imagine that we could make it work with a little bit of optimization.
Excellent. So I believe that's all the time that we have for today for our Q&A. There were quite a few questions that came in that are going to require some additional information, and we will reach out to you directly to the email address that you provided at the time of registration. Nancie, this has been such a great and informal presentation. Do you have any final comments for our audience?
No, hopefully this was helpful and answered some questions, and I look forward to reaching out to some of you with the other questions that we weren't able to get to today. And yeah, again, we're going to be updating the spatial biology page with more resources as we're, developing more app notes, finding out more information about this, and hopefully people find this useful for their research.
I'm sure they will, Nancie. Thank you so much again. And I want to thank our audience for their excellent questions. Now, you can submit questions during the on-demand period, and those questions will be addressed via your email address that you provide. So feel free to continue to drop in those questions. I do want to repeat the websites that we called out today. We have thermofisher.com/fluorophores, thermofisher.com/EVOS1000. So EVOS S1000, and thermofisher.com/spatial-biology. So thank you so much. I do want to also bring our attention to a survey that will pop up as soon as we conclude. We're grateful if you spend a few minutes answering those questions. It will help us learn if we have covered all of your expectations and give us the ability to cover any topics and content related to spatial biology in our upcoming webinars. And additionally, if you would like to be contacted by a sales representative to learn more about Allura spatial amplification kits, click that yes box. I want to thank LabRoots for hosting today's webinar, and Thermo Fisher for sponsoring today's webinar. Until next time, everyone, thank you for joining us. Have a great day. Bye-bye.